Hello, everyone, and welcome to another very exciting seminar in our series. Before I introduce today's speaker, I just want to remind everyone that, as always, there will be a Q&A session after this talk. If you would like to post questions for the speaker, you can do so in our dedicated Slack channel, where you can also upvote with a thumbs up the questions that you would like to see answered. With that, it is a pleasure to have with us today Anjali Goswami, research leader in life sciences at the Natural History Museum and professor in paleobiology at University College London. Anjali's research explores the evolutionary, ecological, and developmental drivers of morphological diversity in vertebrates over geographical time. She uses a combination of high-powered 3D imaging tools, evolutionary modeling, and phylogenetics to analyze skulls, fossils, and embryos from extinct and living species, and also conducts fieldwork across a wide range of countries to understand the major factors shaping organism diversity. Her work is continuing to make important contributions to paleobiology, and she was awarded the Bicentenary Medal of the Linnean Society and the Zoological Society of London Scientific Medal in recognition of her outstanding research impact. Today, she's going to tell us more about reconstructing the evolution of diversity using a phenomic approach. So thank you very much for accepting our invitation, and over to you.
Thank you very much. I'm very excited to be with all of you today and to be able to talk about some of the research that we've been doing over the last several years, and especially about some of the research that's come out over the last year, despite all of the disruptions that we've all had to face. This is really a research program that has been going, that I've been working on for about 21 years now. So it's a long-term project in many ways, and that is slowly getting towards the questions that I really want to understand about the drivers of morphological diversity across deep time. And I think all of us can appreciate that the evolution of diversity is a really fundamental topic in biology. It's important for everything from understanding how we got to the kind of present world with the organisms that populate our world today, as well as trying to project into the future and how those organisms will respond to changes in climate and environment through human activities and other drivers. Now, anybody who thinks about diversity on large scales, as I do, will understand right away that diversity is not evenly distributed, whether we look at space, time, or the tree of life. So if we look across the world today, we know that there are far more species in some areas than others. So there are far more species in the tropics than at the poles, as we can see in this map of species diversity, where there's really hot spots in the tropics. If we look through deep time, we know that there are periods in the past where there's relatively high setting diversity, punctuated by periods where there's rapid decreases in diversity, often associated with mass extinctions, as well as periods where you have rapid increases in diversity, radiations, often following some of those disruptive events. And we also know that if we look across the tree of life, that there are clades that are far more diverse than others in terms of having large numbers of species, large numbers of ecological and morphological diversity. So for example, if we look at birds versus their closest extant relatives, the crocodiles, we can see a really massive difference in the number of species. So what I'm really trying to understand is what processes really shape this diversity and drive these differences in diversity over large scales. Now, all of those examples that I just gave you are actually talking about species diversity. But when I think about diversity, I'm actually thinking about something quite different. And I'm thinking about the diversity of form, or the diversity of morphology. And really, what we can think about is thinking about this in terms of an organism's phenome. An organism's phenome is really the sum total of their observable physical characteristics. And these are really important to think about from an evolutionary perspective, of course, because it's these very characteristics that are mediating their interactions with other individuals, with other species, and with their environment. And so by understanding how these traits evolve, how these physical characteristics evolve, we can understand the factors that are really driving the evolution of these species. Now, as a person who at least considers myself a paleontologist, these sorts of traits are also very important because they allow us to access information that we otherwise can't get from extinct organisms. So things about their ecology and their life history that we can reconstruct from their phenotype, because we'll never be able to observe them directly. And of course, we can take these traits, these phenotypic traits, as well as our inferences about their diet, their ecology, and their life history, and we can build that into a phylogenetic framework to reconstruct the evolution of life on this planet. And it's really important to do that for several reasons. One major one is, of course, we know that the vast majority of life on this planet is extinct. And we know that by building fossils into our reconstructions of how diversity has evolved or how traits have evolved, we can have really radical effects on our estimations of how traits evolve. So for example, if we ignore the vast majority of extinct tetrapods, for example, we'd be ignoring about 2/3 of all of the families that have ever lived and all those morphologies that those represent. Because of course, the second major point is that it's not just that the vast majority of organisms as species are extinct, but that most of the variation across organisms is also extinct. So if we look at just a variety of forms of organisms that we know lived in the past versus those today, we can clearly see that those aren't just simply, straightforward ancestral forms for modern diversity, but rather represent a much broader range of variation than what we can capture from just the modern fauna. And we can also clearly see how these would be very different from what we would estimate as the ancestors of some of our extinct groups. So you wouldn't take birds today and think, if I were to think about what the stem group leading to birds looks like, you probably wouldn't picture, a T-Rex, quite frankly. Similarly, if we even pick something quite familiar, such as sloths, we wouldn't necessarily, if we went back 10,000 years ago, just a blip in geologic time, what we would think of as a sloth is very, very different from what we think of as a sloth today, because the average sloth then were these giant ground sloths, in comparison to the two little arboreal forms that we have today. So bringing this kind of variation into our reconstructions is really critical. Now, time isn't only deep, of course, though. Time also has a developmental axis. And so we also want to think about how developmental systems evolve and change when we try to think about the evolution of morphology. And I think Conrad Waddington put this really nicely when he stated that the evolution of organisms must really be regarded as the evolution of developmental systems. And we can take some of the most obvious examples and clearly see how they would impact our understanding of how morphology evolves. So you can, for example, take the axolotl, classic, very cute animal, and you can take its really close relative, tiger salamander. And if you looked at these two species in their sexually mature forms, and you didn't know anything about how they developed, you might interpret really radical changes in their morphology to allow this big difference in their sexually mature forms. But of course, if you know anything about their development, what's actually happened is a relatively trivial change in timing of development, the timing at which they reach sexual maturity. And so this really radical, apparently radical morphological transformation is actually a pretty simple developmental shift. And so we want to try and incorporate our understanding of these developmental processes into our understanding of morphological evolution as much as possible. So can we really try to understand how difficult or how easy certain changes might have been, which will of course impact on how we reconstruct the processes that drove these changes. Now, ideally, what we would be able to do is we could quantify all of these different factors. We could quantify changes through ontogeny, and we can do that for some species. So we have, for example, in the past, quantified and currently quantified morphological changes through ontogeny, as well as morphological changes across phylogeny, and directly compared those. But that information isn't usually available for extinct taxa. Very, very few fossil taxa actually have much of a developmental record. And it's actually not even largely available for many living taxa. Most taxa don't have a nice embryological series in every museum collection or can't be reared in a lab. And it's really thinking about how can we incorporate this sort of information or these sorts of effects into our analyses that led me to the field of morphological integration. And morphological integration is a topic that is based on a really, I think, intuitive concept that the evolutionary changes that happen in any one trait do not necessarily occur independently of changes in other traits. And by quantifying the variation and covariation of traits, we can reconstruct their correlated changes in variation across species, across specimens within a species, as well as changes across species in a larger comparative framework. And by doing this, what we might find is that all the traits are independent of each other. They're all evolving without any impact on any other traits. Or we might find the opposite. We might find that they're in fact highly interconnected in terms of their evolution. And when a trait changes in, say, the tusk of an elephant, you'll find a correlated shift in its toe bones. Now, what we actually generally find is something a bit in between, where we find there are groups of highly integrated traits, maybe associated with developmental or functional units. And there's other traits that are relatively independent of those traits. And so what we find is that we find these groups of modules, as it's termed, where we have highly integrated traits within a set, but those sets of traits are relatively independent of other sets of traits. And that means that they can vary independently of each other, evolve independently of each other, and potentially then adapt in a way that optimizes their function without compromising the function of other traits. Now, this field of modularity is one that's really been driven by the genetics and developmental world. And only relatively recently has really come into the broader macroevolutionary framework. But it's really interesting and really important to think about for the understanding how morphology evolves for many reasons. Now, one, again, going back to the fact that I'm a paleontologist and I'm constantly trying to think about how we can access more information from just phenotype when we don't have developmental information. We certainly don't have any information in terms of the genetic structure of underlying traits. How can we get at that when all we have is phenotype? And I think a very important point was made in a paper by Young and Hallgrimsson about 15 years ago, that when we reconstruct the covariances of phenotypic traits, and we look at their modular structure or the level of integration among traits, we can identify functional associations that may be driving the selection for that integration of traits. But the underlying target of that selection is development. To produce these high levels of covariation among traits, selection must be acting on the underlying developmental or genetic architecture of traits. And so what that means to me is that essentially when we are looking at the quantitative correlations among phenotypic traits, what we are looking at is the imprint of the genetic, developmental, and functional associations of those traits. So we can get more out of it than just raw morphology. We're actually looking at the architecture that is driving how that morphology develops and evolves. And the other really important factor here is that these covariances among traits are a primary influence on morphological evolution, morphological variation of those traits in individual character evolution. And again, this goes back to this idea that if traits are highly correlated, that means that they're evolving, that they're co-varying in their variation and in their evolution. And if traits might have the way that a trait might be evolving has a negative impact on an associated trait, that might limit its ability to evolve in an optimal direction for selection. Now, because of this influence on individual trait variation, the evolution of these relationships has been a topic of a lot of discussion. And there's this idea that's been hypothesized, and it really, I think, drives a lot of very interesting questions in evolution about whether there are trends in the relationships of traits, not just the traits themselves, but in their relationships with each other. And the general idea that's been put out there is that through the course of evolutionary time, there's a trend towards increasing modularity of phenotypes. And the idea is that by increasing the modularity of phenotype, you'd reduce any canalization or constraints that occur from increasingly complex developmental processes or developmental pathways. And so modularity is a way to circumvent any negative effects of increasing complexity of organisms. And so this idea is that there's a trend towards fragmentation of trait relationships through the course of evolution. Now, the opposite can also happen. Traits can become associated with each other if they are highly associated functionally, and that eventually drives associations in other in the underlying architecture of those traits. Now, which of these actually occurs is an unknown question, and I certainly can't answer it today, but I would hope to be able to at some point. And certainly one that I think is very interesting for understanding really how organisms evolve and how the way that organisms evolve has changed throughout the course of the history of life. Now, to just talk a little bit more about how we think these relationships might affect the evolution of traits, we can use simulations. And the general idea is that trait relationships will limit the variance of traits to specific regions or relatively limited regions of the possible morphospace that they could evolve into. And so here are some really simple simulations just to show this. And these are, on the one hand, mathematically obvious, but on the other hand, I think useful to think about and talk about. So on the left here, this plot that looks a bit like Saturn shows two things. What each of these dots represents an individual, a simulated individual, and the red dots represent those that are evolving with no integration among their traits. So all the traits can evolve or can vary in any direction that they want. And you can see that the end result is that you get this kind of dispersion through the morphospace, this kind of spherical shape. In black are individuals that are evolving with integrated phenotypes. And for this, I just used a standard covariance pattern for a mammal skull. And what's happening here is that the variation across these different individuals is following along the principal axes of variation, which are defined by the covariances of those traits. Now, what you can see here is obviously a whole lot of what is being explored by the red individuals is not being explored by the ones in black. On the other hand, what you can also see, which is a little bit unexpected potentially, is that by forcing all the variation into just a few limited axes, you can actually see that there's more exploration along the extremes of those axes than you would expect without any integration of traits. So what this suggests is that by really pushing variance into just a few areas, you can actually see increased morphological evolution or increased morphological variation than you might otherwise expect without integration. But on the other hand, you also have a lot more individuals being shoved along that one axis, which likely has the effect of increasing homoplasy as well as convergence. And I think this is an important point here because we often talk about convergence as a really unusual or unexpected phenomenon. But actually, given common developmental patterns or shared covariance structures among traits, convergence might actually be more of an expectation than how we usually think of it. Now we can also flip this simulation on its side and look at what's really happening through time. And that's what the figure on the right is. Now in this case, the y-axis is looking at the divergence from the ancestral state. So 0 being the phenotypic state of the ancestor and going up being a divergence from that ancestral form. Again, the red lineages here now are ones that have no correlation among the traits, whereas the black ones are ones with correlations among them. And you can see that the ones that diverge the least from the ancestral form are in fact the ones that are highly integrated. So those are the ones that are, at the 50 time point are still basically at 0 on the y-axis. But again, interestingly, you see the ones that are also the most divergent from the ancestral form are also those that are highly integrated, again showing that by pushing variance across these few axes of variation, you may be constraining it, but you may also be actually facilitating greater evolutionary variation than what you might expect from just completely unconstrained evolution, which is what is in those red lineages. So again, these correlations among traits may lead to more or less change, more or less morphological evolution than uncorrelated trait evolution might predict. Now then, the other question here is whether what we're talking about here is disparity of forms or diversity of form. It's important to recognize that this doesn't necessarily translate to evolutionary rates. So for example, we can think about that sort of axis of variation that as a tube of kind of possible morphologies given a certain covariance structure among traits. And you can think of the evolution of those morphologies as following as like a fly in the tube. So it's stuck within this relatively limited window of possibilities, but within that tube, it can actually zip around fast or slow doesn't really limit how quickly it can evolve. It just limits kind of where it evolves to in that space. And to give this a empirical example, one structure that seems to follow this sort of pattern is the basicranium, or the cranial base of carnivorous mammals. And basically, the cranial base is a region that's often thought of as very conservative. It's really useful for phylogenetic traits, and it seems to evolve relatively slowly, or in a limited manner, we think, compared to, or we usually think, compared to other parts of the skull. And when we measure this, the cranial base is really highly integrated. So there's really strong correlations among all the traits in that part of the skull. And it isn't very disparate. It doesn't actually have much morphological diversity, certainly not compared to other parts of the cranium. But actually, when we measured its rate of evolution, it was actually remarkably high. So what we found was actually, even though it doesn't explore a lot of space, it re-explores that same area of space over and over and over, actually quite rapidly. So it's just re-evolving the same forms over and over. So even if you're locked into a relatively limited area of space, that doesn't actually mean that you can't be evolving quickly within that space, but that effect would actually also increase things like homoplasia and convergence. So we call this model the fly in the tube model of the evolution of integrated phenotypes. Now, so we have these two different possibilities, right? High integration of traits could limit things to staying kind of close to their ancestral state and have lots of convergence and homoplasia, or it could actually facilitate more exploration along these few axes of variation. So the question is, which of these effects actually dominates in the natural world? Because these are, of course, simulations, and simulations can't really tell you what happens. They can just tell you what could have happened. And so that's really what we've been trying to get at through the work in my lab. And some of the questions that we have within that kind of overall theme is, how do the interactions among traits influence the tempo and mode of phenotypic evolution? And also, do these trait interactions reflect things like ecology or life history? So do direct developers have different patterns of trait integration compared to biphasic or metamorphic species and things like that? And are some of these modes more evolvable as a result of that? And ultimately, what we'd love to get at is the question of answering the question of whether there are trends in modularity integration through the course of evolutionary time. So in order to answer these questions, we need data. And so what we've been trying to do is gather a broad suite of data to really try and understand these intrinsic factors that might be influencing the tempo and mode of the evolution of form, as well as trying to look at extrinsic factors and how they relate to those as well. So thinking about things like environment and climate in conjunction with development and genetic influences on how traits evolve, and being able to put this all into a really a comprehensive framework of the different factors driving the evolution of morphology. So in order to answer these questions, we really must quantify morphological evolution and trait integration across taxa. And people have done this. This has been a topic for decades at this point. And overall, people have largely looked more than anything else at mammals. And when we look across mammals, what we actually find is that the overall pattern of skull modularity and integration is generally quite conservative. So we have this kind of straightforward pattern of about 6 cranial modules that we find across mammals. There are some exceptions, especially in the very extreme ecological shifts, such as in the aquatic specialists, such as whales and phocids, seals. But largely, mammals are very conservative in their pattern of integration. And what that means is that gives us relatively limited information or variation to really tell whether differences in modularity actually have any impact on morphological evolution. We need some variation, we need some real differences to actually test that. So what we actually need to do is compare and quantify these patterns in a broader range of taxa. And so that's what we've been doing over the last several years, is gathering 3D scans across the breadth of tetrapods. So looking at amphibians, reptiles, birds, and mammals, to try to put together a broad comparative data set of skull evolution and evolutionary modularity, as well as looking at patterns within species. And so far, this number is probably slightly outdated, but we have about, say, 2,600 specimens that we've scanned, representing 18 to 1,900 species. As I mentioned earlier, we have tried as much as possible to incorporate fossils within this data set because they are important for accurate reconstructions of morphological patterns, as well as for giving us a better perspective on the full range of variation in morphology that's existed. And so far, you can see that within the fossils, they are largely concentrated within a few groups, such as mammals, for example, as well as amphibians, but really are helping to really solidify that picture. And alongside looking across all tetrapods, within each of these groups, we also have multiple species that we have large sample sizes for, so we can also look at patterns within species in terms of trait integration and compare that to the patterns that we see across species, to really try and test whether the genetic and developmental associations that would be driving within species patterns do seem to be replicated at the across species patterns. And so all the people up here in this picture are the ones that have really been driving this research program, focusing on different clades. And so we'll be talking about all of their projects during the course of the rest of this talk. And to gather this data set, they've basically had to travel pretty much all over the world with our various heavier or lighter 3D scanners to collect this data. And that seemed like a really, really difficult part of the project. It certainly took quite a lot of time, but it turns out as soon as you get to the next step of this project that was actually the easy part. Because once we have all these amazing 3D scans, really nice high resolution scans, gathering an incredible amount of variation in morphology, then we have to figure out how to compare it. And that is something that definitely feels like Sarlacc's pit, a new definition of pain and suffering as you're slowly digested, in this case, over the course of 5 or 6 years. And the reason that this is such an issue is that when you're looking at everything from, say, an early tetrapod or a frog to a bird to a whale, there are actually relatively limited comparable landmarks across all these taxa. So when you want to compare the shape of the skull, even though we're talking about the skull, this is a structure that's obviously homologous overall across all these taxa, but the individual elements and the individual shape of those elements is so different that it's extremely hard to find, for example, a single suture point that you can find on all these taxa. But we tried. To start off with, everybody in the team took their clade of interest. They identified all of the landmarks that they could pick out on the skulls. Usually for each clade, on one side of the skull, you might be able to pick out, say, 60 or 70 landmarks that you could identify across all the species within your clade, say all mammals or all birds or all frogs or something. And then we compared our lists across all of our different clades. And the number that we could actually identify across all of these taxa was 12. So across thousands of species, we could pick out with these amazing high resolution data sets, what we could actually pick out was about 12 points that were clearly homologous across all of them. And that's just not a number that does justice to the incredible variation and the incredible morphologies in this sample. Because what we want is to achieve an accurate, comparable, and high resolution description of these phenotypes in order to really understand how these are evolving. And so we took a different approach. And so what we've done is apply something that we think is really getting towards the spirit of phenomics by using a high dimensional geometric morphometric approach. And so to do this, what we do is we take our 3D scans, whether they're CT scans or surface scans, we fix them up, removing any artifacts or anything from the scanning process. And then we place discrete landmarks. So these would be our kind of normal type 1, type 2 homologous landmarks. And we put those on our skulls, and we have those separate ones for each of our major clades. And then we use semi-landmark curves to delineate regions of the skull that are more comparable across all of our taxa. So for example, curves that are outlining the maxilla, even if the individual sutures of the maxilla don't actually line up across our clades. So we end up with something like this. And then what we do is we take this skull with its red landmarks and its golden curve semi-landmarks. And we built a template, and this template is comparable across all of our different species, across all of our data set. And we take these red and gold points and we put them on the template, and then we add these surface points, these blue points that actually describe the shape of the surface of those bones. And so, for example, they would be, in that case, describing the shape of any individual bone that is homologous across all these taxa, such as the maxilla, the frontal, etc. And then we use software in R that we've customized to then take those landmarks and map them from the template onto each of our individual skulls using those red and gold points. And so just to show what that looks like in a bit more detail, we basically are taking this template, using those red and gold points, we are warping it to the shape of each specimen, then taking those surface points, those blue points, inflating them off of the template and projecting them onto our individual specimens. And that gives all of our specimens a comparable surface semi-landmark coverage. And if you want to know the details of how this works, we wrote a very detailed and I think it would be fair to say tedious paper describing each and every step in a lot of detail and showing all the things that can go wrong and how you should deal with them if you go wrong. What are the various considerations you have to take, you have to keep in mind when deciding exactly which element to do or what kinds of options there are, and when you should use one versus the other. So if you're interested in trying to do this with your own data set, I recommend you start there. And then certainly obviously come to us if you have any questions. And once you do that process, you end up with something I think is quite amazing, which is this really nice description of the skull morphology that is comparable across all of our taxa. Now, that sounded like a hard part. That was definitely very hard. But then we get to the next step, which again, felt even harder because once you have all this data, you have to analyze them. And big data sounds great, but big data creates really big problems analytically. It creates huge issues just in terms of the computational power needed. It creates issues with dimensionality. And a lot of the methods that are out there right now are just not built for these kinds of data sets. They're largely built for univariate data when you want to do evolutionary modeling. The multivariate methods that are out there have usually been tested on, say, 10, maybe 20, if you're lucky, traits, but certainly not hundreds. And so we've had to do lots and lots of testing and lots of methods development to try and figure out how to make our data sets work with these different methods. And so we've written some papers talking about that, and I'm happy to answer questions about those, or you can look at some of those papers. And we also wanted to make sure that it's even worth it, which is a difficult question to ask when you're, say, 3 or 4 years down the road in a project that took a huge amount of time to gather these massive data sets. Well, was it worth it? What do you actually gain from having this high dimensional data? I think it's a really important question to ask. And so to do that, what we did is we take these really high density data sets, in this case shown on caecilians and lizards in this figure, and we subsample those data down to their relatively smaller sets of landmarks within each of our regions or each of our bones, essentially. And we also compare them to, say, our normal kind of type 1, type 2 landmarks that you would gather. And we looked at the relative fit of our analyses with the full data set versus the subsample data sets. And what we found is that we are oversampling by probably about double. With about half of the data, semi-landmarks that we have, we'd get about the same pattern or most of the same pattern. But certainly when we're using much, much less than that, and certainly when we're only looking at points that are sitting at the edges of bones, we're getting something that's quite different from what you get from this full data set. And so we think that it is definitely worth it, and you're getting a lot more information from this. One important point is that actually the vast majority of the information may well be coming from the curves for different kinds of structures, but there are certainly groups of organisms that have a lot of morphology along the surface of a bone that you wouldn't get with anything else. So few on that point. And then what you can also do once you have this kind of data is start to compare things that are so different that to compare them seems almost crazy, right? So in this case, we can look at, a theropod dinosaur skull and a caecilian amphibian skull. And these are things that are so incredibly different in their in everything, right? But they are homologous structures. And when we want to think about things like how do patterns of integration or patterns of modularity change across tetrapod evolution, we want to come up with a way to compare these things. And even though their bones are different, and certainly they have different homologous landmarks for each of those structures, they do have largely the same bones in our skull. And so we can take our analyses of those groups, our separate analyses of those groups, and we can compare them and say, okay, do the patterns of correlations across the skull look similar or different? And so we've done this across all of our various groups. And what we find is that, yes, largely there are a lot of differences in how the different parts of the skull are integrated. And those are largely concentrated in the face. So the regions of the skull that actually seem to show really the same pattern, very similar patterns of high integration across all of the tetrapods that we've looked at, which is almost all the tetrapods at this point, anything associated with the suspensorium or the jaw joint region is consistently very highly integrated, regardless of whether it involves a single bone or multiple bones. It tends to be quite highly integrated. The occipital region, regardless of how many parts are associated with it, whether it's one or two bones or multiple bones, also tends to be quite highly integrated. The face can be highly integrated, but usually different elements are highly integrated across the different taxa. So there's a lot more variation in that region than in the suspensorium and the occipital region. And so we are able to take these very different organisms and actually now try to can actually get some understanding of how integration has changed across them. We're still fully mapping that out, but we have the capacity to do that now. And that really answers, I think, a question that I often get asked when I'm talking about these methods, which are quite time consuming, of course, which is why not use an entirely automated procedure? And the reason we don't do that is because we ultimately want to be able to look at the different regions of these skulls and understand how they are related to each other. So if we look at, the maxilla versus the nasal and the premaxilla across these different, all these different taxa, what are the relationships to each other? And that's something that you just can't quite do with completely automated methods at the moment, at least. Although obviously these methods are changing rapidly, and hopefully in the future, they will become much less time consuming. And once we have this amazing data across all of these different species, we can start to get back to the question that we really want to think about, which is, what are the intrinsic and extrinsic factors that influence the tempo and mode of evolution of organismal form? Now, I wish I had time to talk to you about each and every one of the clades that we've looked at so far, but that would certainly take all day. But we've done this across squamates, which was published a few years ago. We have a paper in review looking at this across crocodiles. We have papers looking at birds and mammals that have been published in the last few years. Salamanders. We have papers on frogs this one's actually coming out in exactly 1 week. And our mammal data sets are nearing completion, with a paper looking at the jaw in particular across mammals coming out also in 1 week, and papers on the skulls in prep at this point. So lots of really interesting stuff where we can look at the distribution across morphospace of the different structures. We can look at patterns of integration across these different taxa. We can look at, we can reconstruct their evolution through time and look at how rates of variation have changed or vary across different clades. We can look at what factors, whether life history or ecological, are associated with different kinds of skull shapes or different kinds of rates of evolution. And we can start to draw links across these different clades to see what are the similarities and what are the differences. So again, I'd love to talk about all of these, but I'll focus just on these ones that came out over the last 6 months or so. I'm looking at birds and dinosaurs and at salamanders, because they have interesting stories within each of them. So for a bird data set, this really started off first looking at extant birds, where we took our skull scans representing about 352 species of birds, and reconstructed rates of evolution across that tree. And we wanted to look at a couple of different questions. One, we wanted to look at what factors are really associated with skull shape in birds. And this is because a series of papers in recent years have suggested that diet is not strongly associated with things like beak shape. Now, this is, of course, quite feels wrong, of course, because you could argue that the entire field of evolutionary biology is in some way driven by the idea that bird beak shape is highly correlated with ecology, which, of course, part of Darwin's initial observations. And yet, when you look across the entire clade, And we found this as well, you actually don't find a very strong association. Within any smaller clade you might, but across the whole of birds, there's lots of different ways of being a piscivore or a carnivore and other things. So that seems to hold true, that diet and skull shape are not strongly associated in birds. But interestingly, diet and evolutionary rate are. So certain kinds of ecological groups do appear to evolve faster than others or slower than others. And in particular, it's the nectar eating and the seed eating birds that appear to be quite fast evolving. And that's of course interesting because those groups that are the classic adaptive variations and the classic ones that of course Darwin is interested in actually fall within those categories, right? And so I think it's quite lucky that he wasn't looking at carnivorous birds, for example, or we may not be here right now. We can also look, of course, at integration and modularity and put that into this framework and understand how those factors are related to things like disparity in evolutionary rates, as well as potentially the developmental drivers of those patterns. And so within the birds, what we found was that we do see a huge amount of variation of which parts of the skull are really highly integrated and which parts are relatively loosely integrated. And that's across the x-axis here with the within module correlation, is showing how integrated are each of those within themselves. What we found is that the really highly integrated regions of the skull in birds actually evolve quite slowly and achieve quite little disparity compared to the ones that are relatively weakly integrated. Interestingly, all of those relatively weakly integrated but fast evolving regions have input from multiple tissue types in general, but especially the anterior mandibular cranial neural crest, which shows up in all of them. And so what we suggested is a hypothesis that we can then hopefully test with experimental data and a more focused approach is whether these having this more complex developmental origin of these structures is actually associated both with having relatively weaker integration as well as having maybe a higher evolutionary capacity. And we can also take these patterns and look at in really high detail, where do we see the fastest rates of evolution? And in birds, this is really concentrated in the anterior rostrum. We can also take this kind of data and do fun things like reconstruct what we think an ancestral bird might look like. Now, birds have obviously a really interesting fossil record, but most of it is smashed flat like this example here on the right. And so it can't really be incorporated in our kinds of data sets very easily. But we can reconstruct these in 3D and create an estimation of what we think, or a search image for what we think an ancestral bird might look like. Here. Now, of course, birds do actually have a fossil record in 3D with actually a really exceptional fossil record in 3D, but that's represented by slightly more stemward birds, and those are dinosaurs. And so what we've done now is, because we have this approach that can really compare very different things, we've pulled the dinosaurs into our analyses as well, gathered the same kind of data across the dinosaur skull as we have for the bird skull. So we have the exact same number of data points, exact same positioning of data points across dinosaurs and birds, and well, non-avian dinosaurs and birds. And on these top two figures here, we have our principal components analyses showing the morphospace for birds and dinosaurs. And you can see the silhouettes kind of show the cranial variation that all the major axes are representing. In black are the various birds, and in the colored shapes are various non-avian dinosaurs as well as stem group birds. And what you can see is that there's obviously a lot more birds than dinosaurs in our data set, but the dinosaurs, the non-avian dinosaurs, are actually representing a huge amount of the morphospace variation in our data set. And when we reconstructed the rates of evolution across this entire data set, what we found was that the fastest rates of evolution were observed in the non-avian dinosaurs, several times higher in the non-avian dinosaurs actually than what we see in with the in-prime group birds on average. And we did a lot of different analyses and sensitivity analyses to see if this was driven by things like differences in branch length, differences in sampling, but overwhelmingly it holds under all conditions that really birds are evolving much more slowly than other dinosaurs did. And when we look at all of our individual cranial regions, this also holds true, whether we're looking at all dinosaurs or just the non-avian theropods, or the non-theropod dinosaurs like the ceratopsians and sauropods, birds are actually very slow evolving as far as dinosaurs go. And if you think about it, although we think about birds as this really fast evolving adaptive radiation, this actually makes a lot of sense when you think about the skull. Because within the skull, when you look at other dinosaurs, you see this incredible variation in how they acquire their food, how they process their food. You see lots of chewing and complex mastication across other dinosaurs. You see a lot of other things that they do with their skulls, like signaling with bony tissues, not just with feathers, as well as fighting and other structures. And birds don't really do that with their skull. They do a lot of that with soft tissue structures instead. And really the focus of the skull in birds is really on brain protection. And basically it's a brain case with a beak stuck on it, whereas dinosaurs are doing a lot of other things, or non-avian dinosaurs are doing a lot of other things with their skull as well. And so this actually passes the gut check in terms of making sense, but it was an unexpected result when we got it. And we can also compare that pattern of where the fastest evolution is happening in the skull by looking at how the various different dinosaurs are deviating from the ancestral morphological, or just from the average morphology here. And what we see is that whereas birds are overwhelmingly focusing evolution on their beaks or on their rostrum, you see incredible variation on that part of the skull in birds. But in dinosaurs, you see it actually in other regions, especially things associated with the jaw joint, where you see really fast evolution, things associated with the crests on the kind of parietal occipital region of the skull, as well as sort of the midface. And the dinosaurs that are more closely related to birds may be showing a bit more of a rostrum focused pattern. And so that's something that will be interesting to dig into in more detail. So that's dinosaurs and birds, which are a really interesting study. But actually all this has really shown me, as a person that's actually mainly a mammal focused person, that the most interesting things that are happening in tetrapods are actually within salamanders and other amphibians. And so I just want to close out by talking a bit about some of our work on salamanders, which is led by Anne-Claire Fabre. And again, we gather the same kind of data set shown up here in the corner with really nice high dimensional data across the salamander skull. We looked across, we looked at the morphospace of the skull. And what you can see right away is that the way that salamander skulls are differentiated in shape is really closely associated with how they develop. So whether they're metamorphic or biphasic, more specifically, whether they're pedomorphic, where they stay in their juvenile or larval form as adults or as sexually mature organisms, or whether they're direct developers or viviparous, really discriminates their position in morphospace independent of phylogeny. And what's interesting about that also is that when we look across the salamanders, we find that there's a shift in the rate of evolution consistently associated with shifts in their reproductive strategy or their developmental strategy. So it's clear that in salamanders, that how they develop has a huge impact on how their skull is evolving. Interestingly, habitat actually has relatively little to do with that other than its association with development. So we don't see much of a strong habitat signal within the salamanders, but we see a really, really strong signal for development. And those biphasic and pedomorphic forms are consistently evolving at a faster rate than the direct developers. So those are the blue and the red species here, and the different columns are the different parts of the skull, the different elements in the skull. They're consistently evolving faster. And so we can clearly say that development is a major driver of skull evolution in salamanders. What's interesting about this is that when we looked at frogs, and I'm only going to very briefly talk about the frogs here, and this is again coming out next Wednesday in Nature Communications. When we looked at the same analyses in our frog data set, we found actually the complete opposite pattern. Frog adult skull shape is hugely driven by their adult habitat. And there isn't actually a very strong association at all between their mode of or the development and the shape of the different parts of the skull. Now, in this case, what we've discriminated them as is whether they're actively feeding as larval forms or not, because we expect that to have an impact on their skull function and then on their skull shape. But actually, there's hardly any impact in terms of shape. But what we did find consistently is that when frogs lose the actively feeding larval stage, and essentially become, equivalent to direct developers, where we have a non-feeding larval stage, they consistently show an increase in rate of evolution. So non-feeding frogs that don't feed as tadpoles evolve faster than frogs that do. And so it does seem that actually, in almost exact opposite to what we see in the salamanders, that feeding larval stage in frogs may be constraining the evolution of adult skull shape to some extent. So again, having these having the ability to directly compare patterns within these taxa that have hugely different skull shapes gives us really interesting insight into how development and ecology are shaping the evolution of the skull, but not actually in very expected ways. In fact, we're finding that we see very different patterns from what we would have expected, and very different patterns in closely related taxa. So there's a lot of interesting stuff that's coming out here that will be really cool to dig into in more detail in these clades going forward. And certainly that's something that Anne-Claire is looking forward to focusing on. Then just going back to the salamanders and the topic of integration here, the very last thing that we can see is that these different life history modes also show quite different patterns of integration, where the only ones that don't go through any form of metamorphosis, those that stay in their larval form, actually show a much more integrated skull than those that do go through metamorphosis. And that's, again, something that's really interesting to think about, that going through metamorphosis may actually be associated with increased modularity of the skull. And what that means for evolvability and things like that is going to be a rich topic for future consideration. Now, of course, the last thing that we can do now that we have all of these taxa with the same kinds of data is really compare where is evolution happening across a skull. So whether we're looking at our salamanders or our frogs or our Sicilians, our snakes and lizards and dinosaurs and birds, and we could throw crocs and mammals into this as well, is that we actually see that variation seems overwhelmingly concentrated in just a few regions of the skull. The anterior rostrum and birds, but in most of the other parts of the other taxa, we consistently seem to find that the jaw joint region, or the suspensorium, is where there's a huge amount of variation and fast rates of evolution across all these groups. And so we see this kind of dichotomy across tetrapods and how the skull is evolving. And I think that's something that's going to be very interesting to look at from a developmental perspective with more targeted analyses in experimental systems. So to close, hopefully I've convinced you that this really high density approach is really powerful for comparing evolution across really disparate groups, and for looking at both intrinsic and extrinsic factors on the evolution that influence the evolution of morphological diversity. Now, trait integration is one of those very important factors that can both constrain and facilitate a morphological diversification. And so it's going to be really important to continue quantifying those relationships across more and more diverse taxa going forward to try to reconstruct the processes underlying morphological evolution and how those processes themselves have changed during evolutionary time. Vertebrate skulls are highly modular, but really not all in the same way, although there are some consistencies across the full range of taxa, whether you're looking at theropod dinosaurs or salamanders. And developmental and functional interactions and development and ecology are both really important influences on phenotypic integration and on morphological evolution. But there's a lot of variation actually in which ones of those actually are very influential across major tetrapod clades. And again, not necessarily what we would expect going into this. So we're about 70% of the way through analyzing our data set. So there'll be a bit more coming out over this next year as we start putting the bigger data sets together into more unified analyses. So do stay tuned for that. And we're also developing these in various ways, including developing better methods for looking at trait integration and also for doing high dimensional analysis of evolutionary models. We're expanding to new systems. So now we've moved into pelagic fishes, as well as looking at arthropods and especially insects. And alongside trying to do even more diverse clades, we're looking at how we can do this with more automated methods. And we're also starting to really try to, now that we have some idea of what these patterns are in terms of morphological variation, now we want to try to understand what are the developmental drivers of those patterns. And so we're working with collaborators to really start to interrogate those using developmental genetic analyses, as well as looking at comparative genomic data sets. So all of this is really difficult to do and requires a lot of people thinking really hard and working in a really collaborative way. So I just really want to point out all the people in my team who have really made this work possible and have really shaped how it's gone over the last 6 years now, as well as all of our really wonderful collaborators who share data, share ideas, and share their knowledge about all of these systems. And of course, I want to thank the funders who have really made this possible and made it possible to go around the world and collect these data and really try and figure this out. And especially I want to thank the curators and collections managers across the world that give us access to those collections. Now, as we publish these papers, we are also putting all of our data online. And so if any of you want any of the scans from the papers that have been published, you can go to Phenome 10K and they're all on there to download. And lots of the CT scans are also up on Morphosource. So do feel free to go ahead and grab those and use them for your own analyses going forward. Thank you.
Thank you so much. That was fascinating work. I'm very impressed by all of this, and it's great that you're putting together this database for people to access this. I guess maybe not all labs have access to this high level methodological approach or tools to do this kind of work. So this is really, really great. I was wondering, related to the first part of your talk, where we were talking about examining patterns of phenotypic trait integration and modularity, and you mentioned that mammals are conservative in patterns of integration. And I was wondering if there are specific clades within vertebrates that are not as easy to work with in this regard, and whether looking at also invertebrates is at all possible using similar tools like this?
Oh yeah, absolutely. So I do think that mammals are probably more conservative than most. And maybe that's because we aren't, as a group, actually developmentally nearly as diverse as other groups are. So if development, as I think we've seen in some of the groups, sorry, just got very loud here with helicopters, are with that, with things like if you compare mammals to things like salamanders and frogs and cephalians and squamates, where you actually have a huge amount of different kinds of developmental modes or reproductive modes. And we see that certainly in the salamander data set, at least not necessarily in frogs, but that we see differences in modularity and integration across like the paedomorphic versus the metamorphic forms. Then that could be a huge reason why mammals are more conservative. Now, so I think that's certainly when we look at other groups, they're not as conservative as mammals. There are probably the overall pattern is not so different within those clades. There are the differences across the major clades are certainly larger than the differences that we see within clades so far. So we tend to see, a fair amount of conservation within the major clades, but certainly across the clades we see big shifts. Now, what would be great is if we could actually capture that transition between those clades, but unfortunately, since that relies on having large specimen sample sizes for a fossil taxa, that's quite difficult to do. But we are coming up with ways how we can try to access that. And there are certainly some extinct clades that do have good representation, so we might be able to do that. Now, I think going into groups like arthropods or invertebrates in general would be, fantastic. And so we have started to dive into certainly the insect side of things and mandibulata more broadly to try and look at that. Obviously, there's this whole scale issue when you start to go into clades that are actually really, really diverse. So we've been trying to test out new methods to really access that. But I think what we will find is that there will be much more variation in integration and modularity, probably associated with differences in things like segmentation and how that actually develops across these different groups. There's a lot of interesting hypotheses that are out there, but there's actually really little data available for invertebrate groups. And so I hope that more and more people will start to apply those more difficult clades so we can build up much more information and understanding about how modularity actually evolves across those groups and how it relates to their diversity.
It's very interesting. We have a question here from the audience. What do you think the implications of your work are for studies looking at single traits? Can we understand traits in isolation?
Yeah, that's a really good question. I think we can understand traits in isolation, but I think we need to think hard about how we are interpreting what's the driver of evolution in those traits if we are studying them in isolation. So whether they are actually the target of selection or whether they are actually just a tag along, for example. I think it'll be really important to keep that in mind when you're looking at individual traits, because no trait is actually varying completely independently. No trait is actually evolving independently overwhelmingly. So I think it's, while I think it's completely possible to do, and certainly it's much more straightforward methodologically to do, I think interpreting what that actually means biologically is something that you need to make sure you're considering.
I was also curious if how easy it would be to, in addition to when studying the functional and developmental constraints on cranial evolution, to incorporate data from brain and other soft tissues and see how, for example, brain would constrain cranial evolution across species.
Oh yeah, that's a great question. And there's a lot of people doing a lot of really interesting work along these lines. So some of the people that have been involved with this work, like Aki Watanabe and Ryan Felice, are certainly, and Claire Fabre are all doing much more work incorporating soft tissue into these sorts of analyses. Because I think that is one real constraint of being focused on fossils or trying to get information out of fossils and integrate them into our analyses, is that you are limited to those hard tissues. And what that can potentially mean is that you're missing some obvious linkages across structures, and that you're simplifying potentially some of the constraints or some of the factors that are driving the evolution of those hard tissue structures. So brains are an interesting one because obviously the cranial vault is hugely impacted by brain evolution, especially in things like mammals and birds. And a lot of the change that we see between birds and dinosaurs is almost certainly driven by the changes in brain size. And you can look at how brain development and skull development are related with experimental studies, as people have done. One thing that we're working on right now is actually, and so Aki's work has looked at how the brain and skull are integrated, or is looking at how the brain and skull are integrated across reptiles and birds. And Vera Weisbecker in Australia is also working on this within marsupials in comparison to placentals, and looking at how different developmental strategies may also impact those relationships. So I think there's huge scope to do that. It's methodologically more difficult, not so much for the brain, except for the lack of data from extinct things, and difficulties in actually accurately estimating it. But certainly for things like musculature, you have to worry about things like orientation. But I think that's a very, very interesting frontier for this kind of work, and I hope that there will be much more along that, especially with increasing. All of this is really dependent on increasing access to imaging. So skulls and things like that are really possible because over the last 5 to 10 years, CT scanning has become not just possible, but actually incredibly accessible. For soft tissues, you need to either be staining or using MRIs and things like that. Those will become increasingly accessible, and I think that will really be a game changer for looking at soft tissue integration.
What role do you see for machine learning for these large-scale, high-dimensional phenotyping projects?
Hopefully a huge one. Machine learning is one that we have come back to multiple times during the course of this project, and certainly the insect project that we're getting underway now. I definitely want to find a way to use it for the insect project because that's again a whole different scale from what looking at vertebrates can tackle. The difficulties that we have had thus far is that I think where machine learning has been very successful so far is where you are looking at things that are either highly different, and when you can give it a training data set that can actually identify those things, or extremely similar, and so you're essentially using templates across similar taxa. So it's been very successful for things like mouse systems and things like that. Where it's difficult is where you have things like our data sets right now, where there's some level of similarity, but there's just too much variation to have an effective kind of training data set, at least when we were really trying to explore that as a possibility in our early data collection phases. But I hope going forward, there's been massive improvements in just the last few years, and I hope that will become increasingly possible, especially now that now that we do have much more capacity to provide data sets, training data sets for this, then I think that will become easier. But it's still a little ways away as far as I know.
And we have one last question. When considering how evolvability is shaped by modularity, how do you distinguish faster evolutionary rates due to greater modularity or reduced constraints from faster evolutionary rates due to stronger selection pressures?
Oh, yep, that's a great question. At the scale that we're looking at this at, it's not possible to do that. If you really want to look at that, and really evolvability itself is a question that's, we can talk about it and think about it, but we can't test for it at this scale of analysis. You really need to be looking at population and species level analyses to look at evolvability itself. And at that level, then you can actually start to look at selection, selection pressures, and really model those different effects. But so we can't necessarily distinguish them at this level. The other thing to keep in mind is that when we're thinking about evolution at this level, where we're essentially looking across different genera or across different subfamilies, right? Selection is going to be volatile at those timescales. You wouldn't necessarily expect selection to be consistent across millions of years, certainly, in different and so many generations. So I think the ability to really capture selection is going to be very limited at this scale of evolution of analysis. But what we've been trying to do is look at things at the species level, and also look at things at the interspecific level. So we have a couple of exemplar species. We have things at the across the entire clade, and we can look at whether those patterns of modularity are consistent across those levels. That suggests at least that the factors that are driving the patterns of modularity within species, so the genetic and developmental associations, are actually replicated at that higher level and likely conserved at that higher level. But I think what we need to do is slowly fill in the in-between. So start to build up from the species level up to the kind of cross-species level to really be able to distinguish those factors from each other. So again, we're still at kind of the early phases of building this into a properly comparative analysis across those different scales, but at least we have the fundamental understanding of where is there conservation and where are there changes, and then we can target our analyses to figure that out. But it's a really difficult thing to distinguish those factors. Similarly, I think it's very difficult to distinguish things like constraints versus convergence, which kind of comes back to the selection issue. And those are very, very difficult things to really distinguish at an evolutionary scale.
Well, thank you so much. I think this is all the time we have for today, but thank you so much for accepting our invitation again and for your fascinating talk.
Thank you. It's my pleasure. Thank you for the invitation.
For those of you watching, do join us again next week, where our speaker will be Pat Monaghan from University of Glasgow with their talk entitled Stress Exposure and Longevity, Organismal and Cellular Level Effects at Different Life History Stages. Until then, stay safe and hope to see you again next time. Goodbye.